Welcome to Biochemistry. This video is about purine degradation or catabolism. Purines are double-ring nitrogenous bases, namely adenine and guanine. These nitrogenous bases are found in nucleic acids, which are of two types, DNA and RNA. These nucleic acids are present throughout the cells of the human body. Purines are also ingested through our diet and are hydrolyzed by the pancreatic and intestinal secretions, or they can be catabolized as a part of normal cellular degradation process. The nucleic acids are split into purines and the purines are degraded into uric acid. Uric acid is a waste material of purine catabolism that needs to be excreted from the body. This degradation of purines takes place inside the cytoplasm of the liver cells. Liver is the main site for the purine degradation. Now let's discuss how these nucleic acids are degraded to purines. Nucleic acids DNA and RNA are acted upon by the enzymes, DNA by the deoxyribonucleases and RNA by ribonucleases, and are broken down into oligonucleotides. Eventually, these oligonucleotides are broken into nucleotides by their respective nucleases. These nucleotides are then acted upon by the enzyme nucleotidase, and after removal of the phosphate group, nucleotides are converted to their nucleosides. Nucleoside adenosine is formed from adenosine monophosphate, inosine is formed from inosine monophosphate, and guanosine is formed from guanosine monophosphate. So the nucleases break nucleic acids into smaller units, the nucleotides, and then phosphate is removed by the nucleotidases. In next steps, sugar group is also removed. The nucleoside adenosine is converted to inosine by the enzyme adenosine deaminase. This deaminase removes ammonia from the adenosine, forming the inosine. The inosine is converted into hypoxanthine. After removal of the sugar group, the ribose 1-phosphate, and the enzyme is Purine Nucleoside Phosphorylase. This hypoxanthine is converted into xanthine by the enzyme xanthine oxidase. During this reaction, toxic hydrogen peroxide is also formed. Similarly, the nucleoside guanosine is converted into guanine by the removal of a sugar group, the ribose 1-phosphate, leaving the purine base guanine. Enzyme is the same used for conversion of inosine to hypoxanthine, the Purine Nucleoside Phosphorylase. Guanine is converted to xanthine by the enzyme guanase by the removal of ammonia from guanine. Now this xanthine is converted to uric acid. The enzyme is xanthine oxidase. Hydrogen peroxide is also produced during this reaction. This toxic hydrogen peroxide is then captured by the peroxisomes, where this hydrogen peroxide is converted into water by the enzyme catalase. Now let's have a look at the disorders of purine catabolism caused by the deficiency of the key enzymes. First is the deficiency of adenosine deaminase, and second is nucleoside phosphorylase deficiency. Xanthine oxidase is a very important enzyme responsible for the conversion of hypoxanthine to xanthine and xanthine to uric acid. Role of this enzyme we will discuss later. Now we will study adenosine deaminase deficiency. The ADA deficiency. It is a disorder of immune system. It is autosomal recessive disorder affecting males more than the females. The deficiency of adenosine deaminase results in accumulation of adenosine, as it cannot be further degraded due to the deficient enzyme. This accumulated adenosine is phosphorylated by the kinases and results in the formation of adenosine diphosphate. Further phosphorylation results in the formation of adenosine triphosphate. This ATP inhibits an enzyme, ribonucleotide reductase. This ribonucleotide reductase is a key enzyme that mediates synthesis of ribonucleotides to deoxyribonucleotides, the building blocks of DNA, replication and repair. The lack of this enzyme will result in decreased synthesis of deoxycytidine triphosphate, deoxyguanosine triphosphate and deoxythymidine triphosphate, but will result in buildup of deoxyadenosine, eventually leading to decreased DNA synthesis. Adenosine deaminase is highly concentrated in lymphocytes. Build-up of deoxyadenosine is harmful to lymphocytes, which are dependent on the salvage pathway. As a result, they are unable to grow, repair, and fight infections. Both T and the B lymphocytes are affected, resulting in lack of cell-mediated as well as humoral immunity. This lack of cellular and humoral immunity leads to a condition known as Severe Combined Immunodeficiency. The word combined is for the fact that it affects both the T and the B lymphocytes. The patients suffering from the severe combined immunodeficiency are prone to infections, the viral, fungal and bacterial infections. Treatment of severe combined immunodeficiency is bone marrow transplant or gene replacement therapy, as well as early detection and treatment of the infections. Next is the Purine Nucleoside Phosphorylase deficiency caused by the mutations in the PNP gene. It is a rare autosomal recessive disorder. It is less severe compared to the adenosine deaminase deficiency. Purine Nucleoside Phosphorylase is called the housekeeping enzyme because it clears away waste molecules generated when DNA is broken down. Deficiency leads to buildup of deoxyguanosine triphosphate. The buildup of this deoxyguanosine triphosphate is toxic to cells, especially the immature T cells in the thymus, which damages them and triggers self-destruction. Patients suffer from recurrent infections and neurodevelopmental delays. Now we will discuss the degradation of purines in animals other than humans. Uric acid is excreted as such in the birds and most reptiles. The term uricotelic is designated for those animals that excrete most of their nitrogenous waste as uric acid, usually in urine. Uric acid is converted to allantoin by the enzyme uricase. Allantoin is converted to allantoic acid by the enzyme allantoinase. Allantoic acid is converted to urea by the enzyme allantoicase. Most mammals amphibians, and sharks excrete urea as nitrogenous waste and are termed as ureotelic, excreting the less toxic soluble urea. Urea is converted to ammonia by the enzyme urease. Aquatic animals and invertebrates excrete highly toxic and soluble ammonia and hence are termed as ammonotelic. So this was about purine degradation. This is a part of a series of videos on nucleotides. So make sure to go through all of them for a better understanding. Make sure to subscribe the channel for upcoming videos. Thank you for watching.